So I want to start outside the nervous system and in the gut of all places, where a very common motile bacteria, E. coli, live, and they swim around. And they have a very challenging life. There's competition for nutrients and the environment is in continual flux. So these single cells need to somehow navigate towards nutrient. So how do they do this? Well, back in the 70s, Howard Berg built his own tracking microscope to find out and discovered that individual trajectories of these cells comprise bouts of roughly linear motion interspersed with apparently random turns, and that the tumbling frequency, this turning frequency, depends on the nutrient concentration history. If nutrient is getting more scarce, tumbling frequency increases and vice versa, and in this way, the bug makes its way to a better place. So each bug is essentially swimming around and continually asking itself, much like we do sometimes, are things improving or should I change direction? In short, it is deciding to act or not. And since then, we've learned a lot about how the bug integrates noisy sensory signals, modifies the probability of the flagella motor switching and initiating a tumble or continuing a run, and so on. And there are interesting features of this control system. So some important ones are that there's strong hysteresis in the sensing and motor switching system, meaning that once a decision is made, it is committed. So the bug commits to its actions. Now, this seems trivial, but it's a crucial engineering feature. So essentially, these bugs are swimming around and they have to make these decisions whether to continue swimming in the same direction or to switch. And once they switch, because of the way the signaling is tuned, the switch is committed at least for a period of time. And this has nice engineering features. One of them means it doesn't flicker between running and tumbling. But another one, and this is deeper and perhaps more subtle, is that by committing to a switch and by having hysteresis in this decision, it ensures there's adequate time to evaluate the outcome of a decision. And this is something that maybe is not so obvious when we talk about hysteresis and switching. Now, what kind of evaluation is it doing? Well, again, Berg collaborated with some others and went on to measure the impulse response of the system to a pulse of nutrient. So this being plotted here is the motor bias, which you can think of as the probability that the bug keeps running as a function of time in response to a pulse of nutrient that occurred at the t=5 mark here. And what you can see is that the history positively weights recent signal and subtracts or negatively weights the further distant past, a sort of moving average of the distant past. In other words, it computes a derivative. So this bug is actually performing gradient ascent in this case. So why am I talking about this? Well, it's important to remind ourselves that E. coli is this tiny bag of jelly, roughly a micron diameter. And if for comparison, this is sort of the same volume as a dendritic spine of a typical pyramidal cell. So if E. coli must make these intelligent decisions despite limited hardware, it would be foolish, I think, to assume that a synapse can't, let alone a dendrite or a whole neuron. So the two problems I'm going to focus on this talk are in neuroscience. One of them is the problem that neurons face in integrating signals and deciding whether or not to spike. And the other problem is concerns synaptic plasticity and the situation where a synapse might receive some kind of modification signal, and then it needs to decide whether to essentially remain unchanged or whether to enlarge or modify. And in all of the work that we do here, we try to stick to a few guiding principles. And one of them is that in biology, certainly in the brain, metabolic events are costly, and the nervous system tends to avoid wasted resources. On the other hand, there's a tension here that efficiency, reliability, and performance are all traded off against each other. and none of these things is optimized without a hit to the others. And finally, biology is extremely fine-tuned when it's needed to be. There's a tendency to avoid fine-tuning, but there's plenty of examples where biology is fine-tuned, and the challenge is knowing when that's the case. So in short, biology is basically about things that work, in my view, not just things that happen. They're not natural phenomena that occur spontaneously. And in that sense, and this is the sort of possibly a philosophical debate for the question and answer, biology is actually closer to engineering than physics, in my view. Anyway, let's get talking about the first problem that I mentioned here. And this is the problem that neurons face when they're attempting to perform spiking computations. And I'm going to describe some recent work by my PhD student, Thomas Berger, who's just defended his PhD thesis, and Michael Rule, who's a postdoc, who has just got a job in the University of Bristol. And the question they were asking was whether active dendrites can make spiking computations robust to timing jitter. So before we discuss the solution, we need to outline the problem. And the problem is actually not that obvious to people who've not studied this before. So why are spiking computations difficult? Here is a very, very simple-minded illustrative example. Let's suppose we just have one single neuron. And this the single neuron's job is to decide whether an input stimulus corresponds to a cat, or let's say a dog. Okay, so there is some binary input that the neuron receives, and it needs to either reach threshold or not, depending on the binary class of the input. Now, if the inputs, which in this case are spikes, arrive to the neuron at the right inputs within the right time interval, then there's sufficient time for the neuron to integrate these inputs, reach threshold, and then spike if it needs to. And it can deal with a little bit of jitter, and a jitter within a sort of single membrane time constant will obviously integrate and won't cause a problem. However, fairly obviously, if the jitter and the timing interval between these spikes is randomly dispersed over several time constants, then obviously it's far less certain that the neuron will in fact spike. And this is the essence of what makes spiking computation difficult. And obviously, as the jitter increases, the peak depolarization will decrease, and the variability in the firing rate of this neuron will increase. And if one was to try and combat this by adding additional inputs, then the inputs needed to combat this jitter scales quite badly. So errors accumulate at single neurons, and in a network, this is even worse. So the errors will simply propagate in the network. And to illustrate this, Thomas took a spiking classifier, which is just a multi-layer perceptron, based it off of a standard sigmoidal rate-based perceptron, and trained it to solve a binary decision task. And the binary decision task is simply an abstract classification task where there's multiple classes, and this classifier simply needs to decide which classes the input stimuli come from. And they're being displayed here in a sort of two-dimensional cloud, but just as a technical point, this is a binary classification problem. So the input features were binary features as used in the task. So Thomas proceeded in a very naive way just to illustrate the problem. He trained a standard rate-based or sigmoidal binary classifier to solve this problem, which it does very well, and then simply naively transported across those weights to units with the equivalent spiking rate probability that were actual spiking units. And unsurprisingly, the classifier does very poorly, particularly when the firing rate, the average firing rate, is low. And in this case, the firing rate is simply a Poisson modulated rate. And as the firing rate increases, then obviously the performance starts to approximate the rate-based network. But very, very high rates are needed to reliably approximate the rate-based binary computation. So either one can increase the rates, or equivalently increase the number of units that correspond to each of the blobs in this network. So one can increase redundancy in this network and essentially average across a population of identical units. And again, the scaling is pretty poor. So to achieve any sort of reasonable performance, we need 100 copies of each of these noisy neurons to do anything. So this illustrates some of the problem. There are many, many good proposals for dealing with this issue. I'm not going to describe all of them because they're very smart and they're actually quite diverse. And it's likely in my mind that the brain uses many different methods to solve this problem, and it will depend on the brain area and the exact problem being solved. Instead, in this talk, I'm going to talk about a possible fix to this problem, a very generic one that simply captures some key input-output properties of actual real neurons. So here is a plot showing what the input to a pyramidal cell, so an excitatory pyramidal cell, how an input to the dendrites affects the membrane potential of the soma. And it's fairly well known by now, and work over the last 2 decades now has sort of established this quite firmly, that for sufficiently strong input to a dendrite, it's possible to trigger a large, long-lived event called a plateau potential, often associated with the activation of NMDA receptors. And this plateau potential contrasts very strongly with the typical subthreshold excitatory postsynaptic potential seen here. The property that many people have focused on has been the threshold-like nature of this nonlinearity. But essentially, it gives a yes/no answer to whether there's sufficient input to each dendritic branch. And somewhat less attention, I would say, has been paid to the duration of this plateau. It is associated with events that contribute to synaptic plasticity and other kinds of metabolic events. We're going to kind of ignore those in this part of the talk. We're simply going to focus on how this affects the input-output function of the neuron. And the thing I'm going to draw attention to is that the duration of this long plateau is conspicuously longer than the duration of a sodium action potential in an axon, for example, hundreds of times longer. And that should arouse suspicion. Why does the nervous system need to hold on to these events for such a long period of time? A period of time which, again, suspiciously sort of corresponds to reaction time and these sort of longer sort of intermediate timescales in neurocomputation. So we set our attention on this phenomenon, and what we wanted to do was qualitatively capture the dynamics of this nonlinear dendritic compartment. So we took an existing study by Garrett et al., who had done these nice recordings of single pyramidal neurons and then stimulated dendrites directly by puffing glutamate to measure the response. And not only that, they'd actually used this data to build a semi-realistic biophysical model by reconstructing the morphology and attempting to parameterize this model with the relevant ion channel densities and so on. Huge amount of work, and we are simply piggybacking on the back of that. We just took that model as is and used it to probe the input-output properties and simply ask, are there nice features of these dendritic transients that can enable spike computations to function correctly despite this timing jitter? And the main feature we were interested in was, can we reliably get these nice dendritic plateaus, and can we trigger these plateaus on different branches? The answer is we can, and this shouldn't be surprising because this was one of the main properties that this paper was trying to capture. So using this model as a kind of template, Thomas developed a simplified model where there's a somatic compartment and then these electrically coupled dendritic compartments, which have a stylized sort of nonlinearity in here. So there's a simple dynamics of each of these compartments where there is a threshold, and once that threshold is crossed, then the compartment becomes active, and then that activity dies away and decays after around 50 milliseconds. And what's being shown here is how the inputs to these compartments and then the output of the soma compares to a simple point neuron, which has a simple passive membrane. And here we can see the summation that's occurring through the active compartments causing a spike. And if there's sufficient temporal separation between these inputs, then obviously they decay away and they don't sum to the same level, and you don't necessarily get a spike. And you can probably see where this is leading, okay? The dendritic compartment is this useful short-term memory that kind of performs something which in engineering we call a zero-order hold. It gets a binary input signal and kind of holds it at whatever that value was, so that it can be converted into an analog signal. Now, the hold is only for a short duration here, relatively speaking, but that duration is often long enough to capture the typical jitter that you would see in spike times. So here is, for comparison, the voltage traces in the biophysical model, and you can see that qualitatively this captures the gist of what the model is doing, but is far more tractable to work with in a network. And this is what Thomas used to solve the problem. So going back to this binary classification problem, Thomas now had the problem of constructing a network not out of standard binary neurons or integrated via neurons, but out of these hybrid neurons with these dendrites. And this is the basic structure of the model, where each layer now consists of inputs that go into the dendritic compartments before going onto the somatic compartment. And essentially what this allows each of the units to do is gather input from the dendrite over an interval of around 100 milliseconds, and therefore tolerate jitter in the timing within that window. And this increases the robustness of the network in a classification problem quite dramatically. So essentially, as long as the timing jitter doesn't grow as large as the duration of the transient, then the spiking computation remains intact. Importantly, this does not place a limit on the speed of the computation. So as long as the spikes and the input arrive within a narrow enough window, then the output spike will be triggered. On the other hand, if they take longer to arrive, then essentially the dendrites wait for them to accumulate. So this, if you like, puts a kind of lower limit on the reaction time of a single pass of a computation, but not an upper limit on the speed, if you like. So in summary, the duration and the binary nature of these dendritic plateaus make them a nice engineering solution, which we could describe as a zero-order hold to spiking jitter. And there's a few notes. So this mechanism obviously requires sufficiently strong inputs. Inputs that do not trigger these dendritic spikes would not benefit from this mechanism. It doesn't imply that the computation is rate limited by the duration of the dendritic transients. On the other hand, loading successive computations through the same circuit obviously would require those transients to decay, but it's possible that requirement is not a serious problem for the type of computation that happen in cortical circuits. And we don't make assumptions about plasticity pathways, which are obviously something strongly related to these sort of NMDA related transients. And this mechanism might be part of that picture. Here, we're instead focusing on how to propagate and preserve an input-output transformation through a spiking network using some known biology. Okay, so that is very quickly the gist of this first question. So now let's focus on this other question, which is still in neurons, but it's a very, very different problem, and it's occurring in a very different timescale. And this is the question of whether or not a synapse should commit to change its strength or not. And I'm going to start by revising a very old idea, and that is that learning results from modifying connections in the brain. So if we have a synapse, some kind of plasticity signal, which could be encoded in the activity of the neurons or a modulatory signal or something else, triggers modification of the strength of the synapse. And that, for excitatory synapses, tends to correspond to a change in the volume of the synapse. So the postsynaptic density will increase in volume if the synaptic strength increases, and vice versa. So the synapses can enlarge and potentiate, shrink, depress. New synapses can grow and existing ones can die away. And importantly, there is a limit, empirically, there simply is a limit to the size of a synapse. And it's an interesting quick question, what enforces that limit? Again, a fairly classic idea here is that there's some type of biochemical switch underneath this process. So this plasticity signal, which could be very brief in duration, occurs. And then there's a kind of trace. So some sort of biochemical change happens in the synapse or in the dendrite or the neuron that records the fact that something has happened and some changes need to occur. And during this trace, the synapse weight starts to change. And this process is extremely complex, involved biosynthesis, receptor insertion, structural remodeling, and a whole bunch of other things that happen. And it kind of makes sense that this trace should be reasonably long-lived in order to allow these changes to occur, but be triggered by an event that's really punctate in time. So here's a schematic of this. The plasticity signal will trigger some kind of switch, that will flip on, and this will trigger growth. But how are the switches formed? Right? And how can they function in a noisy, small biochemical environment like a synapse? A very classical proposal is that there are certain molecules that do a good job of this. One of them is a protein kinase called CAM kinase 2, which has this curious capacity to be able to autophosphorylate itself. So once it's been activated or turned on by a signal at the synapse, it can keep itself on and its neighbors on if the signal is strong enough. Okay, so it has this nice switching property. And a classic model going right back to the 80s by John Lissman predicts that there should be hysteresis and the activation of CAM kinase 2. So as a function of calcium concentration, calcium being the relevant variable here because this is triggered by synaptic activity and coincident synaptic activity in particular. As a function of calcium concentration, the net phosphorylation rate of a population of CAMK2 enzymes can flip from being negative to positive. Okay, so for a population of enzymes that are initially off, a sufficiently large calcium transient will cause this net positive rate of phosphorylation, and then this causes positive feedback, which spreads through the population and turns them, many of them on. On the other hand, if the enzyme is already on, then the calcium needs to dip to a much, much lower level to change the net phosphorylation rate to a negative value and thereby pull down the population to this average off value. So there's this kind of net off and on state, possibly at each synapse, possibly at a larger spatial scale. But the key feature of this is that there's hysteresis. And you can see the hysteresis even here, that the value of calcium to turn the enzymes off differs from the value of calcium required to turn them on. And in that sense, the CAMK2 molecule has an intrinsic memory in it. And this is a very nice idea, and it's been followed and studied for many decades. Here is work from now over a decade ago, showing the phosphorylation of CAMK2 inside a dendrite as a dendritic, as a synapse is being activated by glutamate encaging. And we can even see the activated CAM kinase 2 moving into the dendritic spine as it enlarges, and then the canglions 2 activity subsiding again. So the switch has been fixed, synapse has grown, and then things have returned to their previous state. So that's it. Very nice. End of story, right? Well, it's a bit more complex than that. Unfortunately, if one tries to take kinase 2, as well as its biochemical friends, such as PP1 phosphatase and other important companion molecules, and reconstitute the signaling network in essentially in vitro in a test tube, there's no evidence of hysteresis. Okay, this is really a strong test of whether the CAMK2 system behaves as a switch. So here, in one of these experiments, we see the activation of CAMK2 being plotted as a function of calcium, much like in John Lissman's plots. And there's 2 curves here. One where the population starts in the off state, and as calcium is ramped up, they switch to the on state. And one where they start on, and as calcium is ramped down, they switch to the off state. Now, these 2 curves sit on top of each other. So there's no hysteresis evident, at least this level. Okay? So this means it's not a very good switch. It's simply a hypersensitive threshold. And this issue is complicated. I'm not saying that this can definitively show us that CAMK2 doesn't have switching properties. There is a debate about this, but it's more than 3 decades old and it's consumed hundreds of studies. So what we wanted to ask was what makes a reliable microscopic biochemical switch in the first place? That's how we came to this problem. And this project was led by my former postdoc, Monica, and a grad student in NYU. So in order to attack this question, we stepped back and obtained a generic model of biochemical switch. We have this on and off state, called these X and Y, and we have ordinary differential equations describing and respecting biochemical kinetics, right? In particular, chemical concentrations have to be positive, and chemical reactions have to be reducible to elementary reactions where 2 things bind and unbind and the rest of it. And if you respect those rules, you end up actually with a fairly restricted class of equations that can describe these kinds of reactions, and many of you know this. And as a result, you end up with linear terms and then these other terms described by so-called pill functions. So here we have the rate of activation of this X molecule, if you like, being inhibited by Y and then decaying, and then Y conversely being inhibited by X and has its own intrinsic decay rate. So there's a kind of push-pull between these 2 molecules, or these 2 states of a single molecule, can be thought of that way. And classical theory, if we look at this as a planar nonlinear system, we'd say, well, if this thing needs to be a switch, if there needs to be a stable up, down, on, off state, minimally, we need 2 stable fixed points. And the nice thing about this is that if we play with this parameter m, the exponent, then the null lines of this system curve in such a way that they tend to intersect in 3 places, and you get this nice on state and off state flanking and unstable fixed point in the middle. On the other hand, there are parameter regimes in this same system such that this stable multi-stability vanishes, and we end up with only just 1 stable fixed point. Okay? And that would correspond to the situation here where the steady state activation can potentially change. So this stable fixed point could shift and move around as the values of these state variables or parameters change, but there's only one of them. And the system doesn't get stuck on one trajectory as it moves up or down this curve. Okay? So the single stable fixed point situation corresponds to these macroscopic measurements that were made of CAMK2 behavior. But are these classic microscopic dynamics valid in the case of a synapse? Well, as I've said, they're small, synapses are small, And many molecular species are present at very low numbers, tens or hundreds, and estimates of CAMK2 says there's roughly 80 to 250 copy numbers of the molecule itself in a synapse. And the reactions themselves are probabilistic. So what that means is the formulation of any classical model here, even if you're respecting chemical kinetics macroscopically, shouldn't obey these deterministic dynamics. Instead, you're better off modeling these things using a stochastic discrete model, which obeys the chemical master equation, and where each of the reactions, formally speaking, is a birth-death process, with a certain probability per unit time of making a discrete transition between states. In other words, I can't have half a phosphorylated molecule. I can only have 1 or 0 phosphorylated molecules. Right? But the probability of transitioning between those states is a continuous quantity. Okay, so if we now go back to this model, where we have, in the deterministic regime, we have this single equilibrium, we can replace the exact same model with a stochastic description and simulate it, and that's what we've done here. And what I'm plotting here is the probability density that the system is dwelling in a particular state. And what you can see is this sort of smeared out cloud where the system is hanging out roughly in the vicinity of where this deterministic equilibrium occurs. So big deal. All you've got is a sort of noisy version of this. If, on the other hand, we decrease the number of molecules here from the thousands to the hundreds, here's what happens. This is the same system, right? I'll stress that. Same equations, just reducing the number of copies. We move from a situation where there is a single mode in the probability distribution to 2 very strong modes, okay? Let me just show again. This is the large system. This is the small system, okay? Something qualitatively different is going on here. And it's not simulation, but I promise you that. So what is going on? So in this system, and I've popped the equations up here again, and I've plotted the null points. So these loci where the derivatives of the entities are 0. The discrete system lives on a lattice, okay? We can only have whole numbers of molecules in any given state. And what that means is that the null coins, which still govern the dynamics, because the ordinary differential equations describing the deterministic system are the same equations describing the probabilities of the transitions occurring, right? These probabilities switch direction as the system traverses one single step, okay? So here we have 1 of the X molecules, here we have 0. And the important thing is that these null lines can end up confined, sometimes between 2 grid points, or just a handful of grid points. As a consequence, it's fairly likely that as the system sort of traverses around the state space, it could accidentally hop across these null points, and then hop back again, and so on, and get stuck, okay? So illustrating this in the diagram, if the system starts off in this state, well, it's this side of the null line, so y will start to increase. And then if it spontaneously hops across, y will start to decrease. And the point is that this path can continue for many, many states. And at every state, there's an opportunity to hop across this null line and then start falling back down again, okay? So that means that the system can kind of get trapped for some limited amount of time in this mode. So this kind of phantom mode that's not caused by a classic equilibrium in the system. And then by symmetry, at least in this system, you get another mode in the class. So we have this type of modality, which is like stochastic bistability in small systems. And a nice feature of this is it will get annihilated as the system size grows, okay? What I showed you before was that in the large system, you get a noisy version of the deterministic system. as the system size shrinks, the behavior qualitatively changes, and you no longer get that type of behavior. You get behavior which is switch-like. So if we start off here, we hang out here for a while, okay? And it's not too difficult to tune this system so that the dwell time is arbitrarily long. So you can have an arbitrarily stable switch doing this, as long as you keep the numbers in the system confined within some range. This also generalizes to multi-species systems, and the summary of our paper, which you can read here, it's published 2 years ago in PNAS, is that this mutual repression motif is really important. It essentially forces the dynamics into the negative orthant of state space where it can't live, and it kind of gets stuck, hanging out, it sort of hangs out near the axes, if you like, of the state space, and it's that feature that forms these modes. But going back to this feature of this behavior being dependent on the system size, a nice way of interpreting that, I think, is that the system size itself can act as a negative feedback signal, and this can prevent growth beyond some self-limiting size. Okay, so let's go back and take this system and just imagine, right, each of these modes corresponds to the system being on and the system being off. And let's imagine this is a synapse, right? So if it's in the on state, synapse will start growing. Okay, so we'll draw a threshold for inducing synaptic growth. And now we'll ask what happens if growth indeed happens, right? So as the synapse gets bigger, then it becomes harder and harder for that same synapse to be thrown across the threshold into the growth state. So it self-terminates and essentially becomes monostable in a particular state. So what this means is, as the synapse grows, the system size doesn't need to be sensed. Okay, we don't need some elaborate sensing network. This is something that is just inherent in the physics of the system. It can just disable itself beyond a certain size. The modes collapse, and now the system doesn't behave like a switch anymore. So what we learn from this is that mutually inhibiting bipedal species can acquire this stochastic switch-like behavior in microscopic cellular compartments. So when things are small, when they're confined and they can only interact in a certain number of ways, the stochastic discrete nature of those interactions really matters. And when you take this feature and you couple it to system size, in this case, growth of the synapse, you end up with a feedback loop that can regulate the size of that system. In this case, it's the size of a synapse that's being potentiated. And maybe, not sure about this. This is an experimental prediction, and it's something that would require someone to reconstitute the CAN-K2 system, for example, not in a large bulk material, but essentially in a microfluidic device in a very confined space to resolve this. But it might explain 2-ish decades of these somewhat contradictory findings for physiology. And just because I gave two kind of little stories here, the earlier part of this talk, we learned that dendritic nonlinearities are long-lived threshold-like events, and I think more attention should be paid to that. And as a result, for a sufficiently strong input, they can implement this nice zero-order feature, which enables integration of non-coincident inputs. And among other things, this can make spike-based network computations more robust to timing jitter. So it's one of the biggest privileges of my job is actually working with all these amazing people. And as I said, lots of them are sort of coming and going. So the group I have in Cambridge is really in a period of flux in a good way. They're moving on to, for example, Michael's now moved on to a faculty position at Bristol. Monica has another position here in Cambridge. Thomas, who did the work on dendritic nonlinearities, has just defended his PhD and is looking for other places to go. And I want to thank my collaborators and the rest of the group, because having that group provides a great environment for discussion and critical feedback. And I really want to thank Ran and David for giving me this opportunity to talk to all of you. And hopefully we can have a discussion. It doesn't have to focus on the things I've talked about here. It could be the sort of wider questions and could even stem towards this statement I made about biology being more kind of like engineering than physics, just to try and provoke some of the theoretical physicists who might hang out in this seminar series. So that's the end of my talk. Thank you again for listening, and I guess now we can have the Q&A.